in recent years, there has also been personalized cancer therapy, immunotherapy treatment. And the earlier one, which has been FDA approved, is the CAR T therapy. So CAR stands for chimeric antigen receptor. So these are proteins that can allow T cells to recognize. You can imagine these are almost like antibodies that allow T cells to recognize specific antigen on tumors. For example, all the B cells in our body express CD19 and CD20 on the cell surface, and this includes B cell lymphoma and B cell leukemia. And so there are CAR Ts where the CAR is a T cell, which on the cell surface, the CAR is an antibody that recognizes CD19 and CD20. And so the CAR T therapy works like this. When you have a cancer patient, you have to take their own T cells and then engineer for the T cell to express this CAR, which on the cell surface is this a protein which can be an antibody that recognizes the cancer-specific antigen. And in this case, it will be the CD19 and CD20 on B-cell leukemia or B-cell lymphoma. And then in the Petri dish, we can treat the cell with IL-2. And so the T cell will proliferate. And you can make like 10 to the 10th number of T cells. They all carry this CAR. And then you infuse the 10 to the 10 T cells, like a huge special army, infuse into the patient. And all these CAR T cells will circulate around the body. And anytime you see a B cell that has a CD19 or CD20 on the cell surface, this CAR will recognize it and stimulate the T cell for activation and killing. And so that essentially will kill all the B cells, whether they are healthy or leukemic. But hopefully over time, the body bone marrow will replenish the body with additional healthy B cells, but have the cancer cell B cells wiped out. And so this really has been approved for a leukemia and lymphoma treatment on CAR T, but there are challenges in using CAR T to treat solid tumors. There are following challenges. The first is that we need to create an antibody that specifically recognizes tumor and can spare the normal cells. At least if it kills the normal cells, it doesn't have too bad side effects. So apparently in our body, if we kind of, in a few months, if we lose the B cells, it's not the end of the world. Eventually when it gets replenished, it's okay. And so we need to have an antibody against an antigen that is uniquely expressed on the cancer cells. The second tricky thing is we need to make sure that the T cells can actually get into the tumors. So far, CAR T has worked well in lymphoma or leukemia because they kind of circulate in the body. And anytime it sees a B cell in circulation, it can kill it. And the third challenge is to maintain the CAR T cell activity in the tumors. How do you make sure that the CAR T, once it gets in the tumor, can get activated and kill? For example, the solid tumor can also overexpress PD-L1 and suppress T cell activity. And so you might imagine having the CAR T infiltrate the tumors, but still having to use a PD-1 antibody to maintain the T cell from being drunken by the immune cell, by the cancers. You can see there's a question: do CAR T cells continually attack B cells for the rest of the patient's lives, or do the CAR T cells wash out in the person's blood at some point? They don't get washed out because when you infuse all 10 to the 9s or 10 to the 10s, all T cells into the body. What happens is that those T cells will have over time, they will die. And so it has been shown that sometimes even 1 or 2 years after a CAR T therapy, there's still CAR Ts that you infuse in the patient that are alive, but eventually the cells will get old and it will die off. But hopefully by that time, the cancer cells would have been killed off. And next one is adaptive T cell transfer. So this is you isolate the patient tumor and you separate the cancer cells from the immune cells. And so you grow these tumor fragments in little cultures, and then you try to have the T cells, you can cultivate the T cells and, test them to see which of the T cell clones, you isolate individual clones, which of the T cells clones really recognize the tumor and get activated. And for those T cells, you can expand them specifically. And so in this situation, we only amplified the T cells that can recognize and get activated, can recognize the tumors and get activated. And so you again expand these T cells outside the body and then infuse them into the patient. So this could be, instead of one CAR T, it's kind of like T cell army, and you can have different T cells that recognize different mutations, right? But some of the same problem exist. Here, you don't really have to worry about the antibody because the T cell has receptor to recognize the tumors, but you need to still worry about the T cell getting into the tumors and maintain the T cell activity. Also over time, the T cell will die. There are recently even more personalized T cell immunotherapy. For example, this is from the same pioneer group that does this kind of adaptive T cell transfer. You separate out the tumor and the immune cells. The tumors can then be sequenced for mutations. And so based on the mutations, you can predict which of these mutations might stimulate an immune response. And so there are algorithms to predict immunogenic mutations. And so for those, so supposedly this is a mutation from gene 1, this is mutation from gene 2, this is mutation from gene 3. And so the scientists could create a kind of expression cassette to express all the potential mutated antigens that are immunogenic, and you can create a mini gene, and that includes the predicted immunogenic new antigens, and present this into the immune cells. And so this is usually first presented on the dendritic cells, which specifically recognize antigen that's presented to it. And then the dendritic cell can stimulate the T cells. And then the T cells will have receptors to recognize the dendritic cells. So basically, dendritic cell is a antigen presenting cell, which can present these mutated peptide onto the cell surface, which will then be recognized by T cells, and then the T cells will be proliferated and inject back to the patient. This again is like outside the body, you can create a very special force of those T cells, which specifically recognize the tumor antigens. And once they are ready outside the body, you inject them into the patient. And so depending on the mutations that the patient has, this type of personalized adaptive T cell will have very different T cells to recognize those different mutations. Recently, there are also cancer vaccines. So cancer vaccine is different from flu vaccines. The flu vaccines are mostly preventative, whereas cancer vaccines are usually therapeutic, which means the patient has already been diagnosed with cancer, and after the cancer vaccine, you're hoping the patient will have a boost of immune system to kill the cancer cell. So first of all, in general, viral infection itself, it could be immunogenic. For example, somebody can have a viral infection, whether a flu, or recently there is a case where a cancer patient has not been treated with therapy, but they got a COVID infection, and that boosted their immune system and was able to eliminate the tumor growth. But that's just in general, because the viral sequence contains something that's not in the in our normal body. So once it infects the cells, our immune system very quickly have a boost to say something happened. There's kind of an overall rally of our immune system. But it will be even better if we can create those peptides representing the tumor antigens. So in this case, if we can take out the patient blood and isolate the dendritic cells, but then present the dendritic cells with either tumor lysate. In this case, you can see here, our immune cells can be engineered to present the antigen presenting to present the tumor antigens. So these could be a tumor lysate, could be a viral vector, so it can actually present non-tumor antigens, just virus, or present CpG sequence, just CG-rich peptide CG DNA sequence, which can be translated into little peptides. Or you can have these tumor-specific peptides from mutations to kind of train the dendritic cells that carry these tumor antigens. And you can inject those dendritic cells into the patient, and they will help train your T cells inside the body to really recognize these mutations, right? So interestingly, early days, people use this dendritic cell as a vaccine, but also later on, you can also use oncolytic virus. So these viruses now are vectors. And once they are inside the body, they preferentially infect and lyse the cancer cells because they go to the cells that grow fast. And so cancer cells kind of proliferate fast. So the oncolytic virus will go there, and then they will have a better presentation of the antigens, which will then also boost your immune system. But the really exciting development of cancer vaccine is personalized new antigen vaccine. It works like this. When somebody have cancer, you isolate their tumor and get the DNA for sequencing and call the mutations. And then out of all the mutations called, you can predict which one of them will be immunogenic. And then for all of those, you create a little either RNA or peptides in a kind of a little tube as if it's a vaccine. It's similar to the COVID vaccine from Moderna. It's an RNA vaccine. And so you create a pool of RNA that contain the mutations in the patient tumor that you predict are immunogenic. And then you inject this into the patient. It's like to show the immune system. Here is an example of all the bad guys in your tumor. And so the T cells or B cells or the immune cells in your circulating blood will say, gosh, these are all the bad cells. And they will start to proliferate. And then when they circulate, if there are cancer cells that carry the same mutations, the T cells will know how to kill those. Right? So there are also challenges in here. The first is that usually you cannot give too many. These new antigen vaccines, you can usually only give like 30 at a time. If you give too many, somehow that doesn't really induce a very strong immune response. And so to make sure, from thousands of gene mutations in each tumor, how to pick those 30 that are truly immunogenic, that's important. And also make sure those are presented onto the cell surface to generate an immune response, right? And also, again, how do we make sure the T cells infiltrate the tumors and make sure the T cells don't get inactivated from PD-L1 or from those immune checkpoint? And so these are important. So sometimes the cancer new antigen vaccines are also combined with current PD-L1 treatment. So how does the RNA get into the cells? Yeah, that's kind of the very special technology of Moderna or these RNA vaccines. I'm actually not an expert on this. They have special way to make sure it's, I think for cancer vaccine, you just inject the RNA in and the immune cells will automatically eat this and then chew it up and present it onto the cell surface. But I think for Moderna, there are special ways you can even specify the delivery of the vaccine onto specific cells or organs. And that's something actually I'm not an expert on.